Okay, can everybody hear me? Okay, so let's go ahead and start. Okay, welcome to this session of webinar. I'm Professor Gao Zuren from the PhD program and the International Master Program in Medical Neuroscience. In this session, I will introduce you our programs and hopefully can provide you all the necessary information if you are interested in applying our programs. So neuroscience has always been an important field of research. Medical conditions are related to our central nervous system, such as neurodegenerative disease, stroke, and brain or spinal cord injury are among top threats of our human health. So in needs of effective treatment of this disease, it is thus important or necessary to start to focus on research in medical neuroscience. So with the advantage of Taipei Medical University having 6 hospitals, strong clinical teams specialized in neurology and neurosurgery, our PhD program was co-founded at 2011 together with the National Health Research Institute, NHRI. And also, starting from this year, we also have the international master programs. Okay. So the primary aims of our program are to train outstanding research talents in medical neuroscience and also to develop international collaborations in neuroscience researches. So in this session, we will first introduce you the educational goal and the core competence of our programs. Then we will talk in more details of our programs, including the course information, the supportive resources, including the available space and equipment, and the stipends. And also our faculties. And also we will briefly introduce our faculties and their research. Okay. So there are 4 main educational goals in our programs. The first one is to train scientific talents who can perform research independently. And then we want to integrate the basic and the clinical study and develop translational research. And also we will train students to utilize multidisciplinary technology in medical neuroscience research and the drug development. And then we want them to be competitive internationally with global vision. Okay. In terms of core competence, following the complete training in our programs, the students should first possess the knowledge of medical neuroscience in all aspects, such as nerve regeneration, nerve degenerative disease, and brain injuries. Second, they are able to perform research and operate all the necessary equipment and the technologies, and to analyze research data. Third, they can do literature search and write scientific theses and proposals. Fourth, they can perform multidisciplinary and translational research on their own. And then finally, they can organize and present their own research data and take questions. So to achieve that, we offer a comprehensive coverage of course selection, including all the required and elective courses in several different categories. So here shows the map of course information for our PhD program as example. The course selection for the international master program are quite similar, but a little bit more simplified. So you can use this as example for reference. You will find a broad selection of courses such as neuroprotection, neural regeneration, animal models, research methods, biostatistics, and a lot of other popular topics in medical neuroscience fields. And after you take all the necessary courses, you will have to pass the qualifying exam and the dissertation defense in order to graduate from our programs. And following the study in our program, graduates can select either academic or industrial track to start their career, including faculty positions in school or research institute, postdoc, researchers, or R&D professionals in pharmaceutical or biotech companies. So to allow students to develop their full potential and learn new skills and techniques, we provide rich supportive resources, including generous lab space, as you can see from all the pictures shown here as example, and also culture rooms and microscope rooms. Also, the state-of-the-art equipment are available in our programs and also the core facilities in school. And then the common area in our program allows better access of all equipments and also easier communication among students and also between student and PI. In addition, we provide extra stipend to ensure your study in our program. For example, in addition to school's scholarship, we also provide extra stipends available in our program, up to $10,000 New Taiwan dollar per month. And also, we provide stipends for students attending international conferences, whether they are attending conferences in Asia Pacific region or North America and Europe region. And also we provide stipends for students when they publish scientific papers as the first author during their study in our program. So another key factor of successful graduate training is the mentorship. All of our faculties are not only highly motivated and competitive in research, as you will see in the following slides introducing each faculty's research. So in addition to that, we also care about students and build strong mentorship and bonding with them to ensure their proper development when they are studying in our program. So as you can see from those pictures, at different events, such as during the commencement, after dissertation, and all the casual meetings and the year-end party and the field trips. The overall atmosphere in our program is full of joy and inspiring. So next, allow me to introduce our full-time faculties first, then the honorary faculty. Okay. So first is our director, Dr. Hu Chaorong, okay, whose specialties is in stroke, pain, Parkinson's disease, and sleep and memory disorder. Okay. And the next one is Dr. Li Zhao, okay? Professor Li Zhao, whose primary research interest is in molecular neurobiology, tuberous sclerosis complex, epilepsy, and the biochips. And then Professor Zhuang Jianying, whose primary research interest is in neuroprotection, glioblastoma stem cells, drug addiction, and the signal transduction. Next is Professor Zhou Siyi, who focused on neurocell biology, neurophysiology, neuroendocrine, and neurodegenerative animal models. And then Professor Huang Qizhen, whose specialty is in neurodegenerative disease, neuromolecular biology, and the protein post-translational modification. And then Professor Xie Yizhen, who focused on molecular and the genetic, epidemiology, and the biostatistics. And then Professor Chen Kaiyun, who focused on traumatic brain injury, regenerative medicine, stem cell research, and the translational neuroscience. And myself, Professor Gao Zuren, who focused on developmental neuroscience, exon guidance, signal transduction, and cell biology. And then Professor Ke Qiongyuan, who focused on gene regulation, signal transduction, and the neurodegenerative disease. And then Professor Luo Yijun, who focused on brain imaging, psychiatric medicine, and the speech and the language disorder. And Professor Xu Zongyi, who focused on molecular and the cellular biology, steroidal genesis, glioblastoma, and the physiology. And researcher Wu Liansi, who focused on neurobiology, ALS, and FTLD, and epigenetics. So in addition to the full-time faculties, we also have a huge support from honorary professors, including Dr. Barry Hoffer from National Institute on Drug Abuse at the United States, who focus on research in Parkinson's disease, stroke, aging, and traumatic brain injury. And also Professor Horace Law from the University of Minnesota School of Medicine, who focus on cell and molecular neuroscience, neuropharmacology, and the systemic neuroscience. And also Dr. Senjekun James from Academia Sinica, who focus on gene regulation, neurodevelopment, and neurodegenerative disease. Okay, so next I will present you some videos from each of our faculties to share their research to everyone.
Hi, I am Professor Li. Our lab focuses on a genetic hereditary disease called tuberous sclerosis complex, TSC. This disease causes patients to have benign tumors in multiple organs, and most of the patients also develop epilepsy and may be comorbid with neuropsychiatric disorders such as autism spectrum disorder and anxiety. We use animal behavior tests like novel object recognition test and the open field test, which are frequently used to mimic learning and memory functions and anxiety in humans, respectively, to assess the preclinical therapeutic potentials of certain compounds in TSC. To further study the epilepsies in TSC and the treatments, our lab has established video EEG monitoring system and also working on setting up non-invasive brain imaging modalities to detect alterations in microstructures and functional connectivity in the brain. we further used histopathological approaches to validate the results. We are currently translating our lab results to clinical studies. Our ultimate goal is to develop an effective and better therapeutics for TSC. Thank you for listening.
Hi, I am Dr. Jian Zhuang. In my lab, there are two major projects. One is neurodegeneration study after traumatic brain injury. Another study is drug-resistant mechanism of glioblastoma. For my lab, the first study, we will use several experiments to confirm the neurodegeneration and neuroprotection mechanism after traumatic brain injury. For example, like a 3D imaging, bioinformatics study, and several immunofluorescence, as well as combined with some molecular mechanism research. Also, we will use some clinical sample to verify our result. Another study here, we have a similar strategy like a neuroprotection study. In the glioblastoma study, we also use some bioinformatic analysis combined with molecular experiments as well as with clinical sample to verify. Also, we would like to identify the major mechanism how this kind of malignant tumor is able to resistant for chemotherapy. If you feel interested for our study, you can contact with me or search my name here to directly read our previous study. Also, you can contact with me by email. Also, here I would like to invite you to join my lab. Thank you.
Hi, everyone. I'm Associate Professor Sui Zhou from the Ph.D. program for neuroregenerative medicine at TMU. Now we have a new program for the Ph.D. student, which is the Ph.D. program in Medical Neuroscience, and a program for the International Master student, which is the International Master program in the Medical Neuroscience. The research interest in my laboratory is focused on a chemokine named CCL5, and also the function of the memory formation and the neurodegeneration or neuroregeneration. We found out CCL5 is not only a chemokine, but also show a neuroprotective function in many ways. And we found out CCL5 will promote the bioenergy metabolism, and this function is critical for the hippocampus synapse formation and the memory formation. And related study we published on the molecular psychiatry this year. If you are interested in the memory formation, you can take a look and talk to us. The other is we also use CCL5 to treat the mild traumatic brain injury, and some animal or some people will have some memory impairment after injury. And we found that CCL5 is very critical for the antioxidant GPX1 activation after brain injury. And this function will protect neurons from the oxidative stress after neuron damage. And this will protect animal memory function. Related paper will also published this year, and if you are interested, you can take a look. Besides of that, we're also interested to the function of the CCL5 in neuron differentiation and neurogenesis process. We use both in vitro and in vivo model to do the study, So we also culture iPSC cell to differentiate into neural, or we use virus to treat the animal with CCL5. We can see that the cell treated with CCL5 can differentiate faster than a regular median. And also in the animal brain, we can see more neurogenesis even in adult animal. And besides of that, we like to treat the Alzheimer's disease animal model with the CCL5 and see if they can prevent memory loss or rescue them. So if you are interested in studying neurodegeneration, welcome to join us. Thank you.
Hi everyone, my name is Qixian Huang. Are you interested in neurodegenerative disease? Welcome to join my lab. My research is focusing on exploring the molecular mechanism of this disease. As you can see in the following, most of this disease are caused by the accumulation of insoluble aggregates, resulting from impairment of proteasomes and autophagy degradation pathways. Here, I'm interested in studying the motor neuron disease, ALS. In our previous study, we have explored the comprehensive metabolism and mismetabolism of TDP-43. And we also discovered its ether ligase, ZNF179, that plays an important role in TDP-43 neuropathy. Moreover, we even found out that the Chinese herb medicine berberine could accelerate the clearance of TDP-43 aggregates through activating autophagy system in cell culture model. And in our clinical study, we discovered that exosomal TDP-43 may be a potential diagnosis biomarker for ALS patients. Recently, I'm interested in observing the axonal transportation of 20S proteasome in TDP-43 neuropathy. And these are the experimental models and approach used in my lab. So, what are you waiting for? Come to join my lab.
Hello, everyone. I'm Yichen Xie, an associate professor in Ph.D. program for neuroregenerative medicine. It's my pleasure today to introduce my lab. I was trained in molecular and genetic epidemiology in Taipei Medical University, and my research is focused on stroke. Genetic epidemiology is the study of how genetic factors contribute to health and disease in families and populations, and how genes interplay with environmental factors. As we know, genes alone can cause some human diseases, such as Mendelian disease. However, for many complex diseases like stroke, both genes and environmental factors contribute to not only risk of stroke, but also unfavorable outcomes. From 2010, we initiated a stroke clinical data registry called Formosa Stroke Genetic Consortium. We collect patients' clinical data, treatment data, image data, risk factors, and their follow-up data. We also collect their blood sample, including DNA and serum plasma. Until now, there are nearly 5,000 stroke patients enrolled in our study, and over 4,000 patients are ischemic stroke. Based on our data and biospecimen, we can utilize multi-omic approach to discover novel biomarkers and evaluate the association between biomarkers and risk of stroke and their unfavorable outcomes. Further, we can develop precision prediction model for stroke and its outcome in the future. Here's my recent research papers using genetic data and omics data to discover biomarkers and analyze its association between biomarkers and its risk of stroke and their unfavorable outcomes. Above is my research introduction. Welcome to join us and hope to see you soon.
Hi, I am Kai Chen. I am an associate professor of Ph.D. program in Medical Neuroscience. My specialty are translational medicine of traumatic brain injury, neurodegenerative disease animal model, stem cell and regenerative medicine, and cell and molecular biology. Our research project includes TBI, stroke, and Parkinson's disease. The goal of our research is to develop new diagnostic methods and new treatments for traumatic brain injury in order to improve neurological function recovery after TBI. With our international collaborators, NIH and Case Western University, we have collected more than 700 TBI patients' clinical data, including blood, functional MRI, questionnaires, balance, and heart rate variability. And we find that several potential TBI biomarkers such as ETK-BMS, IGF-1, HRV, and so on. And this biomarker can predict dizziness, depression, and anxiety and sleep problems after TBI. In addition, we develop a new position for all drugs. We find that fencerin, pomalidomide, and GIP can protect neurons through antioxidant and anti-apoptotic effect. Besides, treatment of TBI animal with imodin can restore motor function by reducing the release of glutamate and RS after TBI. In terms of neuroprotection, we explore the neuroprotective effect of exercise, DBS, environmental enrichment, and music therapy on Parkinson's disease or TBI. And we find that exercise can improve motor function and cognitive function of Parkinson's disease. And the mechanism may be through increased expression of neurotrophic factor, BDNF, also increased mitochondrial oxidative metabolism and glycolytic function in the substantial library. As for cell therapy, mesenchymal stem cells are relatively safe and easy to operate clinically. We cooperated with Professor Dermal Zhuang to conduct preclinical trial of genetically modified mesenchymal stem cell for the treatment of TBI. A mesenchymal stem cell overexpression FGF21 increase homing ability and induce neuroregeneration. When MSC FGF21 is injected into the brain of rat with TBI, not only the number of cells migrating to the injury site increase, but also the motor and cognitive function after injury are improved. The number of regenerating neurons also increased. Therefore, MSC FGF21 has great potential to apply clinically in the future. If you are interested in the studies of TBI, stroke, or Parkinson's disease, we welcome you to join us. Thank you.
Hi, dear all. I'm Professor Gao Zuren. I am currently an associate professor in TMU. Let me briefly introduce myself and what my lab is working on. I have my 5-year postdoctoral training in Montreal Research Clinic Institute in Canada. After that, I joined TMU as an assistant professor and got promoted to the associate professor 3 years ago. My primary research interest is developmental neuroscience, especially how neural circuit is established during the development of the central nervous system. In addition, I recently started to study some neurodegenerative disease by applying our finding in developmental stages. Here are some of my recent publications this year. As you can see, most of them are studies related to the neurodevelopment. To talk a little bit more details of what my lab is working on, we focus on studying the molecular mechanism of neural circuit formation by using spinal motor neurons projecting into the limb muscle as our studying model. Until today, we know that in order to establish a functional nervous system, each neuron has to extend their axon to their target cells. This could be achieved by the guidance of axonal growth cones at the tip to their proper targets, expressing either repulsive or attractive ligands in the environment. And the combined effect will drive the turning of growth cones to their target cells. Over the past few decades, a lot of studies have identified at least several families of these guidance receptors expressing at the surface of axonal growth cones, which can bind to their specific ligands expressed in the environment to provide directional information. However, it's difficult to imagine that how this relatively small amount of guidance molecules can establish such complicated neural circuits. It is still important to continue searching for new candidate molecules involved in this context. In my lab, we use both chick and mouse embryos as our animal models to study neural circuit formation. As you will find, many chick fertilized eggs in my lab, which is not a common thing in many other labs. In terms of experimental techniques, we normally started with in ovo electroporation by first injecting different DNA of interest into the neural tubes of chick or mouse embryos and providing currents to allow the entry of this injected DNA into the spinal cord. We then determine whether this injected DNA can affect normal neural development, especially the growth of spinal nerves. So to do that, we can section the limb and the spinal cord tissue and do immunohistochemistry using specific antibodies, or perform retrograde tracing technique to further determine the specificity of neuromuscular connection. Then, in addition to the in vivo study, in my lab, we also do in vitro study by culturing spinal motor neurons into the petri dish and observe their growth using different functional assays, such as the stripe assays here. So this is it for my introduction. If you are interested in my study and have any further question, please don't hesitate to contact me through the following email. Thanks for your attention.
Hello, everyone. Welcome to the co-lab at Taipei Medical University, Taiwan. I am Professor Chong Yuan Ko. My expertise includes molecular biology, cell biology, signal transduction, and neurodegenerative disease. Here are my lab members. My research focus are Alzheimer's disease and glioblastoma. My research interest is the transcriptional factor CCAAT enhancer binding protein delta, CEBP delta. Over the past few years, we focused on dissecting the transcription factor CEBP-D mediated cellular events in astrocytes. including phagocytosis, migration, anti-apoptosis, and oxidative stress response during neuroinflammation in Alzheimer's disease model. We identified different CBP delta downstream target genes and the roles in Alzheimer's disease pathogenesis. In addition to our study of CBP delta, we identified a novel CBP delta downstream target, PTX3, and have revealed details on the biological functions of its transcriptional regulation, especially its participation in inflammation, lipid accumulation, and tumorigenesis. Recently, we also developed the PTX3 antibody for the application in the therapy of inflammation-associated diseases, including neurodegenerative disease and cancer. For the glioblastoma, we also clarified the CBP-mediated mechanism in drug resistance. And further, we will explore more CBP delta-related pathways in glioblastoma development. If you are interested in our research projects, you are welcome to join us. Thank you.
Hi, everyone. I'm Dr. Yujun Luo. I'm an associate professor of Ph.D. program in Medical Neuroscience at Taipei Medical University. Since I am a speech-language pathologist, my research interests include applying different kinds of neuroimaging techniques to the clinical practice. For example, what I'm working on right now is to use EEG equipment to obtain the brain neural activities of toddlers with autism or with developmental delay. And I also investigate their language outcomes, including language expression and comprehension. Taking all the data together, I'm looking for the brain behavior relationship of children's development. My research team is also work on designing music combined language training program for early intervention. If you are interested in pediatric neuroscience research, welcome to join us at TMU. Hi.
Hi, my name is Feng Xu. I'm the assistant professor in the Department of Neuromedicine here. Our lab is focused on studying neurosteroids in the brain and how to target neurosteroids to treat glioblastoma and neurodegenerative disease. And this is my lab member. This one is a home use. So he's happy doing research every day, and he likes to play Pokemon Go. And the next one is showing food. She's very good in finding many cheap things in the website. And this one is a young. She likes to search many delicious food in the internet. This one is also a research assistant, Zhong Han Wang. He's a very good expert in photography. The topic of my paper, including the major topic is neurosteroids in the brain. And we study what is the level of neurosteroids in the glioblastoma in neurodegenerative disease, including Alzheimer's disease and ALS. So welcome you to join us in TMU. Thank you.
All right, thanks for all the sharing of their research from our faculties. So before we end the session, we also have a few words from our newly graduates by Dr. Rami and Dr. Rini.
Hi, I'm Rami from Jordan, and I'm currently a postdoctoral fellow at School of Medicine, University of Maryland. And I'm a proud alumnus of the Ph.D. program in Medical Neuroscience at Taipei Medical University, TMU. Initially, I joined the Ph.D. program in Medical Neuroscience, hoping to understand more of the field of neuroscience and to explore my interest in this field for future career. The program has brought me more than I expected. As I graduate, I can proudly say that choosing to study medical neuroscience at TMU was one of the best decisions I have ever made. Everything taught in medical science starts from the essential basics, so I didn't need to worry whether my background can support my understanding for the teaching materials. Also, the great variety of neuro subjects being offered in the program covered many different neuro topics in the field, together with the organized rotations they offer for students to the affiliated labs and the numerous chances of doing internship in these labs. I was able to explore what I want to do in the world of neuroscience and where my true interests lay. I realized that my interest in medical neuroscience has flourished during my PhD thesis work. This eventually led me to do a postdoctoral fellowship. So this is where I am right now, as a postdoctoral fellow at the School of Medicine at the University of Maryland. Looking back on my days in the PhD program of Medical Neuroscience and at TMU, what I found unforgettable is not only the neuroscience studies that I did, but also how nice and warm and welcoming and caring people that I met in the program and in the school throughout my study. That led me to grow as a person. So now I'm more willing to speak up and be brave for presenting my ideas for discussion. This all due to the kind encouragement, inclusiveness, and respect that I received from my teachers during my PhD studies. I'm very thankful to them for making my experience in medical neuroscience and in TMU such as a happy and lively memory that will always stay in my heart. Studying medical neuroscience and in TMU is the best decision I have ever made. Thank you so much.
Hi, my name is Renia Joy, and I recently graduated from the Department of Medical Neuroscience from Taipei Medical University, which we call in short TMU. So why I chose TMU is because it's one of the top 400 universities according to Times Higher Education, and its campus is very friendly to international students, and they offer interdisciplinary courses in which they teach the courses both in Chinese and as well as English. So it allows you to broaden your horizons when it comes to picking up an area of expertise in your research field. Also, apart from that, TMU has diverse and experienced group of researchers, educators, clinicians, and business professionals. So this allows you to develop a very strong network, which can be useful when you graduate and when you're looking for a job after your PhD or master's. TMU basically focuses on clinical oriented translational research, and they assemble interdisciplinary research teams from institutions worldwide. So that can be an asset to you while you're looking for a job. Now, when it comes to my particular department, that is the field of medical neuroscience, the curriculum focuses on neuroscience, cell therapy, and neurodegenerative disorders. So the research areas are, our department is also in collaboration with the NHRI, that is the National Health Research Institute in Taiwan, and the courses as well as the resources are shared between TMU, NHRI, and also additional labs that we collaborate with. So this actually broadens our horizons and broadens our opportunities when it comes to choosing our area of research or our clinical experiments. We also cooperate with well-known institutions around the globe, and we jointly develop the treatment options and or help in the improvement of conditions that are related to the central nervous system. So considering all these facts, I would like to stress on the fact that If all these factors cannot ensure a bright and prosperous future in your career for you, I do not know what can. So considering all that I've told you right now, I would like you to think about it and make an informed decision about joining TMU and how TMU can help you in making a brighter future for yourself. So all the best. Thank you.
All right, this is it for this session. If you have any question, we are having a session of chat and meetup at December 20 and the 21st between 10 and 12. So we are open up for any discussion and take your questions during these two sessions. Okay. And then for most of the information and the latest updates you need, including recording of today's session and also applications informations, you are welcome to visit our program website. Or if you have any further questions, you can email our secretary, Yuling, or you can email me directly if you have any further question. Okay. Thanks for your attention and hope to see you soon. Thank you.